Hello, this is class Bio 225, Genetics, from Genes to Genomes. We will start Chapter 8 today. This is class for Chapter 8, Part 1, with Dr. Lucy Abgarian. Chapter 8 is about what genes are and what they do. Today's class particularly is going to be about genetic code. We'll cover some early experiments, what happened during history of this science that led to discovery of genetics, gene coding, and also how the transition is happening from DNA, how information is passing from DNA to RNA and then to protein. Let's talk about four basic themes that are the keys for flowing the information from DNA to all the way to protein. First one of them is obviously the pairing of complementary bases, and it is the key for transferring the information from DNA to RNA and then from RNA to protein. Second one is due to polarity, directionality of DNA, RNA, and polypeptides, that information is being read properly. Because, for example, in DNA we have from 5' to 3', so there is clear direction how the information should be read and then translated, and that's helping to have a proper gene expression. Third one is energy. So the gene expression itself, it's a very energy hungry process. All the sub-processes during gene expression requiring very high energy. So here comes to help a bunch of ATPs in a cell system that are very high with energy, and they are helping to for functionality of all the enzymes that are taking part in the process of DNA translation, RNA to RNA, and then taking the RNA to ribosome, and then synthesizing the polypeptides. All of these processes are very high energy processes. That's why there are ATPs and other macromolecules within the cell system that are made to support these processes by providing high energy. And the fourth one, the fourth theme would be the mutations. And we know that mutations can change the genetic information, and it can have, from previous classes we have seen already, that it can have a dramatic impact on phenotype. However, it was a key process for genetic as a science, for understanding the whole process behind it. So it's one of the most important parts of genetic science. Here is a quick review chart for showing the flow of genetic information from DNA to protein via RNA. So RNA polymerase transcribes DNA to produce an RNA transcript. Then ribosomes translate the messenger RNA sequence to synthesize a polypeptide, and then the translation is following. The translation of the genetic code is following afterwards. So here comes again, we have talked about it previously, that individual amino acid is encoded by triplet codons of nucleotides. So we have 4 nucleotides, and by 4 nucleotides you can have 64 codons basically. 3 codons are signifying the stop sign, and it's very essential for protein sequence to have start codon and stop codon, so that when the reading is happening, enzymes know where is it starting. Is it a synthesis of new protein, or are you just continuing to synthesize the previous protein? So, and where do you stop? When are you done with synthesizing the protein? So that's why the stop codon is very essential. And then for start codon, there is not particular start codon. However, methionine is known as a start codon, besides being an amino acid that can be within the gene sequence as well. So ATG or AUG will encode the methionine. So as you can see, we have 20 amino acids, but there are 61 codons that are representing these 20 essential amino acids, which means that some of the amino acids can be represented by multiple codons. In some cases, 2 amino acids can represent the same I'm sorry, 2 codons can represent the same amino acids, or in some cases 3 or even 4. For example, in case of leucine, you can see that 4 amino acids are 4 codons are representing the same amino acid leucine. What does it mean? That means that the third nucleotide is ubiquitous, which means that if there is a mutation taking place in a DNA sequence, if the mutation is happening with the third nucleotide, that means that in final product of our protein, it will not affect. You will not see a mutation in a protein sequence. It will still be coded as leucine, because leucine is CUU, CUC, CUA, and CUG. And in another cases, like there are 2 amino acids that can be, 2 nucleotides that can be switched the third ones and then still have the same, for example, in case of histidine, and so far and so forth. So this is very important to, nobody's expecting people to memorize the how each amino acid is encoded, but it's very important to be able to use this information and to be able to know where to look for this information and how to read this information. First, it was Charles Janowski who basically suggested that there is a connection, there is a relationship between nucleotides and amino acids. So what he did was that he generated large number of tryptophan minus auxotrophic mutants in E. coli. So let's look at the detail, like steps that that it was conducted by. So we know that TRPA is encoding the tryptophan synthetase. So he did genetic mapping of TRPA genes, and he was doing it based on recombination, and we know already how that's done. And afterwards, he determined the amino acid sequence of mutant tryptophan synthetase, and that's when he saw the correlation between the nucleotide sequence and between nucleotide synthesis and the amino acid sequence, and that's how he came up with the idea that there is a relationship between those two. Here is the schematics of that experiment that he conducted. So he knew the genetic map of the polypeptide, and he knew where the mutation was located within the polypeptide. So this is by introducing different mutations of nucleotides within the sequence, he discovered that more than one nucleotide is involved in uh, encoding of one amino acid, and also he discovered that each nucleotide is responsible for only one amino acid. It's involved in designing of one single amino acid, and that's how, by putting all of this information together, he conducted that uh, what was the relationship between amino acids and nucleotides in genetic information. So how it was discovered that it was exactly three nucleotides that are encoding the amino acid, a single amino acid. So it was shown by Crick and Brenner in 1955 by experiment. And for experiment, they used bacteriophage T4, and they used proflavin, which is an intercalating molecule, which means it's a molecule that can bind, that can be inserted in a DNA strand. And by doing so, it so basically intercalates into DNA, and also it can cause insertions and deletions by getting there. This is how the experiment go. So first they treated the DNA by proflavin, and they introduced a mutation, which was so one mutation, and then they did treatment of DNA again by proflavin the second time, and they got the second mutation. And when they looked, apparently they still had the restoration of the wild type function, and the question was like, why is that happening? So when they looked at the recombinant uh, uh, DNA, recombinant version of the DNA, they saw that both mutations are still there. So what's happening? What was the case? So what was happening was that one mutation was a deletion, another was insertion, and both of them were shifting the sequence, the entire genetic sequence, one way or another, and they were compensating for each other. So, what was the conclusion from the experiments that were conducted? The conclusion was that codons must be read in order, and very importantly, they have to start from very significant, certain start point, not just randomly from anywhere. The start point in nowadays is called reading frame, the beginning of the reading frame, the origin of the reading frame, and also the introgenic suppression occurs only when wild-type reading frame is restored. So let's look at this little table. So this is an interesting fact, which was basically leading the scientists towards the discovery of the three nucleotides reading one amino acid. So when there is one insertion or one deletion, we are dealing with mutant. When there is two insertion or deletion, we still have mutant. If there is four insertion or four deletion, we will get mutation. If there is one insertion and one mutation, we will get a wild type. If there is 3 mutation, or if there is 6 mutation, I mean not mutation but insertion or deletion, or if there is 3 insertion and or there is 6 insertion, with all of these cases we are getting wild type. So which suggests that the encoding is happening in triplicates. So we will have a deletion of whole amino acid. However, we will still have to restore the rest of the gene sequence. Now let's look at that schematically. So here in first line, we have an example where we have one insertion of C and one deletion of A. So as a result, we are getting a shift of sequence. We are having, instead of GCG, we are ending up having CGC. And then we are having GCC instead of CCG, and we are having GGA instead of GAG, and then we are having GGA instead of GAA. So of course that part of the sequence is going to be mutated. However, the rest of the DNA sequence is going to stay complete without any mutations. So the second scenario, where we have 3 deletions of 3 different nucleotides at different that are part of 3 different amino acid code. So T from AAT is gone, C from CCG is gone, and A from GAG is gone. Again, that fragment is definitely being mutated. So instead of AAT, we are having AAG, and instead of GCG, now we have CGC, and instead of GAG, we have GGG. So, of course, that part is being mutated. However, the rest of the DNA will remain intact. So, and then the third case, when we have insertion of nucleotide, G is added to GCG, T is added to CCG, and C is added to GAG. We are having another type of shift, widening of particular sequence, and we are ending up having this little fragment GGC, GCT, CGG, CAG, which is going to be mutated, and again rest of the DNA will still stay complete. Now let's look at a single addition or deletion of nucleotide. As you can see, first case is a deletion of T from AAT, and because of one single nucleotide being deleted, and for reading one amino acid, we still have a triplicate, so AA ends up being read with G from the next amino acid, and everything is getting one nucleotide shift. Therefore, the rest of the DNA is being messed up. So we are, what we call here is that we are having a reading frame shift. And the same thing is happening in case of insertion. So we have G inserted in GCG sequence. So again, it's inserted in a triplicate within the triplicate. So, but still, the amino acid is going to be read by 3 nucleotides. So instead of GCG now, it will be read by GGC, and the G from original sequence is going to move to the next amino acid reading, and so far and so forth. So that's why we are dealing again with a frame shift here. So since the discovery, there were some technological breakthroughs that happened between 1950s and 1960s. So one of the big ones was discovery of mRNA, the messenger RNA, and also the translation of synthesis of messenger RNA outside of the cell, meaning in vitro. And for that, first, it was involving cellular extraction preparation that was going to allow translation in a test tube instead of cell itself. And then there was a technique that developed to synthesize artificial RNAs for a known nucleotide sequence. And then with all of the above mentioned taking under consideration, it allowed for synthesizing of very simple polypeptides. In 1950s, studies in eukaryotic cells led to discovery of the RNAs, the messenger RNA particularly. And so how that happened, it was so there was an evidence that the protein synthesis takes place in a cytoplasm, right? So there were experiments where amino acids were tagged with radioactive molecules, and it was by the experiments it was clear that there is an intermediate process happening, molecular intermediate between the nucleus and protein synthesis in the cytoplasm. So that's how scientists discovered the messenger RNA that was playing the role of the transporting agent for genetic information. So this is one of the coolest experiments conducted back then. So in 1961 by Marshall Nuremberg and Henry Mathai. So what they did, they had an extract of cytoplasmic extract basically, which contained everything basically would have the cell for genetic information, and they added artificial messenger RNA into that system, and what they got was that they ended up getting polypeptides that was synthesized in that tube. It was radioactive. It was radioactive because the nucleotides in it had a radioactive tag. So basically, this was a clear proof of messenger RNA being the tool, being the mediator for translation of genetic information from nucleotides to nucleotide sequence to polypeptide stage. So after all of these experiments, here is what was shown, that when one takes synthetic messenger RNA poly-U, then the synthesized polypeptide is going to be all phenylalanine sequence. If the mRNA is poly-C, then it's all going to be prolines. If it's poly-A, it's all going to be lysines, and if it's all G's, then it's going to be glycine. So as a result, not as a result, but then the next experiment was with poly-UC, with alternating uracil and cytosine, and then also they did with AG alternating nucleotides, and they did with UG, and they did with AC, and they got as a result polypeptides that were also all being alternating. and then they did a triples, UUC, AAG, UUG, UAC, and they were getting three different amino acids synthesized. They were getting phenylalanine, serine, and then leucine. Or they were getting lysine, arginine, glutamic acid, and so forth. And then when they did they did also the quadruplicates, when they had uracil, adenine, uracil, uracil and cytosine, and they were getting now 4 alternating amino acids as a result being synthesized in a polypeptide. So this was another way also showing that each of the nucleotides is being translated, each 3 nucleotide is being translated into 1 amino acid. In 1965, Nuremberg and Lehrer conducted another interesting experiment. So at this point, there was some information about transfer RNA already and about ribosomes. So ribosome was added to a reaction in one where there was a labeled transfer RNA serine specific that was labeled with radioactive label. And it was introduced to the synthetic trinucleotide, and also another case was with leucine specific transfer RNA, the same situation. So then after the reaction, after mixing this together, this entire reaction was led to flow through a filter, and the filter was specifically to bond with ribosomes. So if the ribosome if the transfer RNA ends up binding the ribosome, then we were going to see radioactivity in the membrane. Otherwise, the radioactivity was going to be in a flow through the waste, basically. And in this case, we can see that that was the leucine, and which suggests that there is a complementarity between the transfer RNA that and also the messenger RNA that were involved in translation of particular amino acid, in this particular case of leucine. So, as a conclusion, today we learned that genetic code has triple codons. Codons are non-overlapping. There are 3 nonsense codons that don't encode an amino acid, and those are UAA, UAG, and UGA. Genetic code is degenerate. Reading frame is established from a fixed starting point, which is called the origin of the reading frame. And codon for the translation initiation is AUG in case of RNA and ATG in case of DNA. mRNAs, messenger RNAs, and polypeptides have corresponding polarities, and the mutations can be created in 3 ways: frame shift, missense, and nonsense.